welcome everyone to the AlphaFold webinar series. So we had an excellent first webinar on scope vision of AlphaFold. And today we are going to discuss a bit more about the impact of this amazing development in structure prediction on teaching and training in life sciences. So we want to understand how we might change the teaching training or where the gaps are, etc. So we have some excellent speakers today, and the whole webinar will be chaired by Alexandre. So he's the first speaker as well. Alexandre is at Utrecht University and works in computational structural biology and has many services and has developed software for prediction of protein complexes and many other things. So over to you, Alexandre.
Thank you very much, Samir, for this introduction. So before I start showing you a few slides I prepared on the topic, I want to introduce shortly the panel that we have today. So after I will start, I will kick off, and then as second speaker, we'll have Ezgi Karaca from the Izmir Biomedicine and Genome Center. Ezgi is assistant professor there. Our next speaker will be Panagiotis Kastritis, who is a junior professor at the University of Halle in Germany. And the last speaker will be Dina Schneidman, who is assistant professor at the Hebrew University of Jerusalem in Israel. So they will all present, we will all present our own views on the impact of AlphaFold on teaching. And we hope to have a lot of input from the audience as well, because our views will be very narrow, most likely depending on what we're doing. So as Samir told you, I'm a professor of computational structural biology at Utrecht University. I belong to the chemistry department at Utrecht. And actually in the past, I've also been the director of chemical education. So next to teaching, I've been managing the teaching and teaching developments at Utrecht University for chemistry department. So what are we teaching in Utrecht? Among others, of course, chemistry is a broad topic, but under chemistry, we are teaching structural biology in general. And my personal interest is in the structural biology of interactions. So students get to learn the experimental methods like NMR, X-ray crystallography, cryo-electron microscopy. Those are the methods that give us, say, full atomistic descriptions of molecules. And next to that, we also teach various other biochemical or biophysical or spectroscopic techniques that allow you to study biomolecules in general. That's the experimental side of things. But of course, if you want to study structural biology and the proteins and the biomolecule, which are at the core of everything that happens in our cell, we also need to rely these days on computational tools. And some of the tools that I'm teaching in my course are related to homology modeling, molecular dynamics, docking, which is, say, also the research topic that we are working on. And when I'm presenting these slides, usually more at, say, research conference or workshops, I'm adding now, of course, that when it comes to interactions, DeepMind and AlphaFold multimer is playing a role there and an important role in the future. But let's concentrate on teaching. So in terms of teaching, just to put the landscape a little bit, I'm teaching at two levels in our university. I'm teaching a second-year course for bachelor students on NMR and molecular modeling. This is for both chemistry and molecular life science students. And I'm teaching a more advanced course in our master molecular cellular life science on structural bioinformatics and modeling. Actually, both courses have quite some overlap, but the levels and the topics that are handled are quite different. And next to that, I'm also contributing to various other courses and PhD school. So what is it that I've been teaching for many years now in Utrecht? When it comes to modeling the molecule of life, you first have to start with models and the force field. So how do we describe interactions? How do we describe particles, atoms? How do we describe the interaction between those? This is coded in a force field. So how do we calculate energies, basically, of a given structure? And then we need optimization techniques to either go from the experimental data to the final model or to the modeling, pure modeling. And many of those are dealing with finding minima in a complex energy landscape, so energy minimization techniques, molecular dynamic simulations, all kinds of practical aspects of running these kind of simulations, and also all kinds of oversampling methods. For example, at the master course, I'm also speaking about Monte Carlo approaches. And of course, homology modeling, and what is also very important is a bit of structure validation, because especially when you're doing modeling, the computer might always give you some kind of model, but it doesn't mean that the model makes sense. So learning to assess those structures is very important for students as well. And also teaching about modeling interactions, which is the field of docking. And in these courses, I'm giving lectures. There are exercise sessions where people might be deriving things on paper or using some web portal to do some simple analysis. And in the advanced course, we also do pretty much half of the course consists of computer practicals, where people are working using web portals, but also the Linux command line to do simulations. Now, one of these practical that we're running, which is part of the master course, and you can find the link here, so it's available online, you can follow the practical on your own, consists of 3 steps. So we start by doing homology modeling of a protein. So we use the MDM2 P53 complex for that, and we model I think, the human protein sequence from the mouse one. So this is say classical homology modeling. We use Swiss model. Of course, Swiss model these days points to the AlphaFold model if they are present, but we still want students to go through those steps. And then since the other component is a peptide, we have the students run molecular dynamic simulation of those peptides and try to do all kind of analysis, clustering of solutions. And at the end, we combine the result of those 2 steps, so the homology models that were generated with the peptide conformation that we extracted from molecular dynamics to do docking using our software ad hoc. Now this year, so this keeps the students busy for about 2 weeks, half of the time, where they have to learn many new skills. So some of the students have no expertise with Linux when they start this course, and they learn a lot. Now this year, we added a new module. So we added a bonus module at the end of the course, which is the AlphaFold 2 module, where the students actually using the same system that they have been modeling, say hands-on, just use the sequences, use AlphaFold to do the modeling and get results. Now, we have only made this module appear at the end of the course because this is actually working very well. So they get very nice models. And I guess that I would be afraid that if we give them that at the beginning of the course, they will be kind of disappointed or not motivated anymore to go through all the classical modeling and analysis steps that we are teaching them. So this module is also available online on our website. Now, next to that, I'm also using AlphaFold these days in a second year short practical that we do with second year bachelor students. So they have an 8-week project where they are doing that about 50% of the time. So we are speaking of students who might have followed some of the basic modeling courses that I gave, but that are usually they will have no expertise at all of Linux. And this year we attracted them with an AlphaFold project, asking them to actually model. You might recognize the picture here. You might see part of an antibody here. So we asked the students to basically assess the capability of AlphaFold to model antibodies and concentrating on the analysis of the hypervariable loops. So that's something that's very easy to do for them. They don't need any expertise in terms of computing of Linux. They can use the Jupyter notebook. They use a bit of PyMon for their analysis. So it's pretty low level in terms of complexity. And they might use it even without knowing really the magic behind AlphaFold. So a few thoughts now about AlphaFold in teaching and the impact of AlphaFold in teaching. And this is purely my personal view. So I think that it will be important, and this will remain important, that students, especially in chemistry, structural biology, life science in general, that they need to learn the principle of structures. This is key. So they need to be able to look at a model and understand maybe that there are problems with it. So that's will not disappear from the teaching that we are doing. Now, if we think of Samir mentioned, all the sequence might have a model by the end of the year. To learn structure, to assess structure, validate structure, what we have in a PDB is in principle perfectly fine. So we don't need the EBI AlphaFold database to do this kind of teaching and learning. It's great that it's there, but it's not a requisite. What AlphaFold is bringing, I think that there are some new aspects there that are valuable, because if you look at the PDB, of course, you only find pretty much the ordered regions of proteins, except if you look at NMR entries where you will see these odd tails. But AlphaFold models the complete sequence in principle, and then you might see that for some entries, there is more than 60% of the sequence is actually disordered regions. So this is something that can be kind of visualized now, and this is something that can make the students more aware of the complexity and the dynamic range that we have in proteins. So that's I think that's something new in a sense, and valuable. I think it will still be important, especially while I'm teaching in chemistry, that the students still understand the chemistry and physics beyond the 3D models. So they should understand how do we get to those. Structural biology is not going to disappear. The experimental methods will still be there. So for them, I think it will also be important to understand those methods, also understand how modeling approach are used to transform the experimental data into 3D models. So all the topics that I'm teaching to the students, it's not only modeling or purely modeling, but it's also methods that are used in combination with X-ray data, in combination with NMR data, in combination with cryo-EM data to get the structures that we see in the PDB. And remember that everything in the PDB is a model to some extent. There's, I think, no single structure that was calculated purely from data without a little bit of model. Now, what makes the success of AlphaFold, I think, in teaching? Is it AlphaFold itself? It can be very easily included in EG. I gave you examples. So we put it in a master course, but we also put it in a second-year bachelor course, and students can deal with it very easily. What is critical here in terms of success factor for teaching, in my view? I think Jupyter notebooks are here the critical thing, or one of the things. So it makes it very easy to use AlphaFold. So all the notebooks that are provided by DeepMind, but also many from of Schneidman. So the AlphaFold GitHub repository is really valuable for that. So that's something we can point students to, and usually they have no major issue to start using it. So that's one thing. But I think AlphaFold will not have the impact that it has, both in research and teaching, if there were not computational resource associated with it. So I think Colab, so that Google is making freely available computational resource, and you get GPUs associated with that. This is fantastic. It's easy to use. You don't need Linux in combination with Jupyter notebook. And I think that's the real impact for teaching, in my view. So if Colab was not there, I'm not sure that we will be introducing AlphaFold to students. Then I think the impact will remain very limited. Simply, I'm sure that many of you have installed locally AlphaFold, then you realize that you need 2 terabytes of space just to put the model. Now, don't expect a student to have a laptop that can host 2 terabytes of space. Don't expect to have computer rooms at your university that will hold 2 terabytes of space. I think that's all. If we had to run the models on local resources, this will be very quickly impossible to run. It will mean that we'll have to run them on HPC system within the university, and then that brings back all the complexity of accessing those resources, knowing about Linux command line level. So AlphaFold is fantastic, but it's fantastic together, I think, with the Jupyter notebook and Colab. So that's, I think, my view of the impact. So we have to see the entire ecosystem together, which also means that Colab might also have a big impact to teach other computational tools to students. With that, I'm going to close my short presentation. So thank you very much. You can find all the online resources about the tutorial that I've been discussing at bonvinlab.org. And I'm going to pass the screen to our second speaker, Ezgi Karaca.
Okay, thank you, Alex, for the introduction. Also, thanks for the invitation to present our view on how AlphaFold impacts our teaching efforts, in particular life sciences. So I will today discuss my perspective on teaching with the use of AlphaFold in molecular biology and genetics departments, mainly because I'm a faculty there. And occasionally I also give like short lectures in medicine department as well. So my experience is mainly based on this level of teaching, and I usually teach at the master's and PhD level, so I don't teach bachelor level courses. So the department that I'm in is a very new department, so it was basically established together with the inclusion of my colleagues, and I was there the first one offering the computational structural biology course, which of course gave me the freedom to design it myself, so that was great. But on the other hand, it was at the beginning, it was a bit challenging for me, and I guess also for my students, because I'm coming from an engineering background and I have been trained in the computational field, like modeling field, for a long time. And because of this, I've never had the view of molecular biologists, basically. And that's why it took some time for me and my students until we align with each other. We speak our own language. But in the end, as Alexandre was introducing, the idea in our field is to be able to link sequence structure and function at the fundamental level by the use of fundamental knowledge, which comes from physics, chemistry, or mathematics. So I've been teaching this course at the basic and advanced level for 5 years, basically for 10 semesters, and it has changed quite much over the semester. So, and it's in a more simple shape now than I anticipated initially, because I realized that my students, they did not have much knowledge at the structure and function level. So their knowledge was mainly, or has been, or is mainly accumulated at the sequence level, which is very precious, of course, but it's very hard to change the perspective from there to structure and function. And by using the sequence level, obviously, they're still interested in understanding the functionality of the proteins that they're studying. For example, they would like to understand the impact of disease-causing mutations, or they would like to understand how a specific target is, how a specific protein is targeted with a drug, or how the protein is functioning within an assembly of complex. But of course, understanding these from sequence is very hard. But on the other hand, just giving all the background, both in structural biology as well as in modeling, in a single course is very hard as well. So based on the experience and with the discussions and with the reaction from my students, I realized that to try to teach all of these ground level information, basically coming both from modeling and protein or structural biology fields, it will be too much to digest over a single term. And as I offer an elective course, there hasn't been also an opportunity to offer a follow-up course so that this knowledge can be distributed over multiple terms. And the result, what I ended up with was teaching them more structural biology and less modeling, less computing, basically, just to make the best use of the time. As I was at this position, then AlphaFold came in by the end of 2020. And obviously, as we all know, it arose a huge curiosity and interest, not only in the community that we are together right now, but also in microbiology students as well, basically in everyone, because this was in media everywhere. So in the end, by having this impact of this tool on the media and at the research landscape, I also experienced something that, like, my lecture became out of a sudden, not out of a sudden, hopefully, but more cooler than before. So this already gave me an extra energy to update the lecture content that I have. And what I usually now use is when I'm talking about a specific protein or specific system. Earlier, I was refraining to choose systems for which there is no structure available. And I even found it very hard to explain to students why a certain part of the structure is not resolved and why we have only the structure of a subset of the system. So considering this, having the resources, having UniProt and AlphaFold's protein structure database resources, this has been extremely useful to me. As Alexandre was mentioning, for example, if we check the protein structure or the model that is being studied by one of my students, let's say, now we can go to the UniProt website during the course and check the disordered part and really explain to them what is physically meaningful and what is not physically meaningful by checking at the model. So basically, with these resources, I now have the opportunity to have also the image of the system that these students are interested to work on. And I also have the opportunity, especially with the UniProt database, to link the variance and function information together with the structure, the whole structure prediction that is out there. But expanding on this, of course, I still would like to address the points of the students where they would like to understand the impact of disease-causing mutations, etc., etc. And for this, I prefer not to use any extra prediction tool. I prefer to teach them how to visualize the structure and go over them together, so that through this, they will have a better understanding of structure and they will learn more structural biology in this manner. And this, I guess, also aligns partially what Alexandre was mentioning, that even though these tools are available in the end, everybody would still need to read and understand what a structure means. So this is these resources are at the monomer level, and if we want to address something at the complex level, we use the CollabFold of AlphaFold. I mean, there is the DeepMind version as well. I don't think that it matters that much for teaching, but we have been using this, and we continue using this resource as well. And based on my earlier experience, for example, earlier I was refraining to teach them docking because, as I said, it would have been too much for them just for one single semester, because in docking or in a complex modeling, there are multiple steps and there are decisions given at each step. And because of this, you need to teach also a certain level of background and etc. And this takes obviously some time. But with this sort of tool, at least within the concept of teaching, now it's very straightforward to make a prediction, to make a complex prediction in a short time, which would provide some more time to teach more structural biology. And with this, for example, I mean, this is at the preliminary stage, but we have prepared one GitHub page to teach how to use CollabFold to predict a complex interaction. And this we have already taught in a workshop, and it went quite well. But again, in this case as well, the content is quite concise. But now there will be, thanks to this, maybe there will be more time to discuss about the biology of the system. And finally, while I finish, I would like to also discuss very briefly on my future perspectives on this topic. I think with the speed of the structure prediction tools that we have at the moment, now we have an opportunity to go beyond structure prediction during our classes, and so that we can teach individual aspects of the points which are of interest to our students. And for this, I guess we should need to discuss further what to put in for how to make this as sustainable training materials. And before I finish, I just would like to also let everyone know that maybe some of you already know that the latest new CASP round is on right now. So by the end of this year, maybe we'll hear some more exciting news from this community. Let's see how that goes. And with this, I would like to thank everyone and also the attendees, and hand the microphone back to Alex.
Thank you very much, Ezgi. So now we're going to switch to Panos. We go from Turkey to Germany.
Perfect. So thanks a lot for the invitation. Basically, a lot of the things in my presentation were previously said by Ezgi and Alex, but I can also show you my personal perspective. So here in Germany, I am at a university of 21,000 students, and yeah, I am a junior professor since August 2018 at the Institute of Biochemistry and Biotechnology. I am heading the cryo-EM here, and I got my positive evaluation for the permanent position. So the teaching, the research, and the committee work that I am doing was judged positively by the university. And I'm heading a lab that we do both research, but also teaching as a lab. So, and where do I teach? I teach at the Bachelor in Biochemistry, as you can see here, at the Masters in Biochemistry, and I am training PhD students And in particular, in the bachelor in biochemistry, I am teaching experimental physics where I cannot see AlphaFold being integrated, but I'm also teaching in biophysical chemistry, and I'm also having bachelor students to do their 12-week rotation in my lab. So in biophysical chemistry, basically, we are teaching fundamental things like amino acids, their properties, and also like the protein structural hierarchy from primary structure to quaternary structure. And basically, in the lab, what we are doing is that bachelor students, they get familiar with Bimol, and by giving them, like, for example, amino acid propensities from Zhu and Fassmann and all of these kind of seminal papers in secondary structure and correlation to amino acids, they can build their own secondary structures in Bimol. And this is quite nice because in Bimol you have hotkeys that you can learn basically the single letter code of the amino acids. And also you can choose different folds and then start visualizing, explaining the students about the hydrogen bonds. And I was very happy actually when I saw on Twitter from Sergey his tweet. This was an amazing actually idea that Sergey had, that basically he developed a minimal version of AlphaFold, where basically now the students can use AlphaFold not for predicting proteins, but connecting these kind of secondary structures. And I'm going to integrate it for sure in the next semester. So on top of like learning how amino acids are, how are the hydrogen bondings in secondary structures, on top of that, to visualize by putting their own amino acids, what kind of folds you have. So it's quite nice. And for the bachelor thesis, we have a complete workflow in the lab from model organisms that we have to like getting assemblies of protein complexes. And for the bachelor, we expect that the student gets familiar with one of these kind of methods that we have in the lab. And yeah, for the bioinformatics, we didn't have a student yet that wanted to do computational structural biology during these last 3 years as a bachelor. But the idea is to integrate what Alex showed and what Ezgi showed for teaching them on a case-by-case basis in my lab, in my systems. So for the master's course, I am teaching in the structural biology and bioinformatics module, and I'm also accepting students, master students, for 24-week duration rotations. And here I am teaching a 10-hour module in cryo-electron microscopy, and I have a laboratory exercise that includes model building in cryo-EM map. And as I said here, it would be great to integrate this kind of bonus modules that Alex showed. And also now that I saw Ezgi's things, maybe also integrate those, but I am a junior professor there. So I'm one out of the 4 people. And yeah, like it's always a matter of like time, how much time is allocated in the course to teach the students. So yeah, it's something that I was thinking about. Anyways, here you can see like a video where we are providing a high resolution cryo-EM reconstruction that we have from the lab of apoferritin, and the students put there the model and they start like seeing what was wrong with the model. Here, what was wrong, as you can see, was a different amino acid that was not fitting very nicely in the density. They are trying with CUT to start like optimizing it and seeing that this is not the correct amino acid. They try a different amino acid to fit. And by putting the correct amino acid, the tyrosine there, then they can see that it fits very nicely, and they can start connecting the atomic models to like lower resolution data. Like this is the idea of this project, of this teaching. And actually with AlphaFold, because we don't have the water molecules, it would be great if some of the students use AlphaFold models and see that there are a lot of densities that are missing, and there are more biophysical principles behind protein structures that can be modeled and understood, like hydrogen bonding with structural waters and all of this cool stuff. So In the master thesis, it's the same principle as the bachelor thesis, but now we expect the students to go through more protocols than one that we have in the lab. And we had basically a master student in the lab who collected cryo-EM data, reconstructed the map, and did with AlphaFold that Ezgi and Alex showed, the molecular models to fit them in the map. So it was also a learning exercises how to interpret a low-resolution cryo-EM map. And actually at the PhD level, here in Halle, there is a weird situation that students are in different graduate schools and they have different sources of funding. So it's a bit like heterogeneous, the students that I have in their background. And one of my students, Yannis, like he was self-motivated to learn AlphaFold. He contacted himself like Gregor Chonowski in EMBL Hamburg, who is an AI expert, and together his cryo-EM data and models, they basically developed an AI pipeline to understand like this kind of densities that we have of protein complexes directly from native cell extracts. I'm not going to go through the whole protocol, but basically Yannis integrated models from AlphaFold. He saw which model fitted in his map, which model didn't. Two models fitted equally from two different proteins. And then by extracting the backbone and finding the correct sequence with another AI from Gregor Chonowski in EMBL Hamburg, he could basically make a very nice model that fitted the experimental density at 4.5 angstrom resolution. And actually, you can see here that these PhD students that are like self-motivated, they can make very beautiful models and start understanding themselves what is correct and what is a bit more like an imagination for the models. Like, for example, here the AlphaFold model showed this kind of backfolded alpha helices in the structure, but in our experimental data from full complex endogenous complexes, we see that this helical density is completely absent. So this is a disordered region or unfolded or flexible region in the endogenous state of this particular molecule. Then also, like other AlphaFold models, again, they had folded domains of the sequence directly interacting with this kind of cubic core. And we can see that this kind of fold is again absent in our densities, although it's biologically and functionally relevant as a solution that AlphaFold gave. So it's not like abundant in the cryo-EM data and our class averages that you can see diffuse density here. So basically, the students can also like start imagining with AlphaFold what it is like to produce structures like Yannis did in this cover. And and the last part that I wanted to stress is also what Ezgi and Alex stressed about disordered regions. This is also another conceptual thing that AlphaFold can be used. For example, here in Halle, we have a graduate school on intrinsically disordered proteins. I have a student there and two connected students, and basically they start learning the functional, the functional aspects that could stem from like this kind of disordered regions and unfolded regions. And AlphaFold database is perfect for that, for visualization. So the students can see all of this kind of manifest disorder in the protein models, and actually start using the AlphaFold database to see where the disordered regions could be possibly. And of course, this is not actually predicted disorder, but we know that it's some kind of proxy, right? So it was quite cool as something to add on top of our graduate school, which has to do with disorder. So overall, it's my final slide. I think that teaching of biochemistry and structural biology is influenced by AlphaFold at all university levels, especially for understanding the hierarchy of protein structure, primary, secondary, tertiary. AlphaFold can play a very good visualization role. Also for the model building, it could be quite important to understand like what is not there. And also it impacts the thinking process for protein structures. For example, as I showed you with the example of cryo-EM of endogenous protein complex from native cell extracts, and about flexible regions of proteins and their interactions. And thanks all of you. That's it. These were my thoughts. Yeah.
Thank you very much, Panos. So now we switch to our final speaker of the day, but not least, Dina. The floor is yours.
Hi, thank you. I'm teaching a structural bioinformatics course in the Hebrew University, and I can say that it keeps changing. So I'm teaching the course with Barak Raveh, who is also a fan of integrative structure modeling. And our audience is students in undergrad computational biology program. But we also have students from computer science and engineering programs who have no background in protein structure. So we start with introduction to protein structure for those students who are not familiar with that. And then we cover structure basics, structural alignment, folding, docking, integrative modeling, and simulations. Our focus is very algorithmic because all the students have computer science background. So we want them to code and implement. So for example, they implement basic structural alignment using triangle superposition. They implement the Markov chain Monte Carlo simulations. They write code to cluster structures and so on. So overall, they have 5 programming exercises. And we used to do a final project, but for the past 3 years, we are doing a 2-day hackathon. So hackathons are very popular. So the last week of the semester, which was last week, we are sort of booking them a room, a computer lab, and all the students gather there for 2 days. We make sure they get pizza, and they work on their projects. So deep learning is right now one of the 5 exercises, so it's about 20% of the course. Okay, so how do we do it? So I'll start with the lectures. So we started with contact and distance prediction, which was what folding was about 2 to 4 years ago. And that was relatively easy to explain because mostly they relied on convolutional neural nets. Most of the students we get are after IML course, Introduction to Machine Learning, and very few have done Introduction to Deep Learning. I'm also teaching them C++ course, and there I make sure they implement the convolutional neural net without training, but they do understand the concept. And then now we have Rosetta Fold and AlphaFold, which have more advanced architectures. So this year I have added a whole lesson about the language models to explain them how Evo-Former module works. And the structure module is relatively easy to explain because we do cover reference frames in the structural alignment part of the course. So this is useful. So we try to explain the details, but not too deep. And now the challenge is how do we do the exercises, right? We cannot send them to implement AlphaFold. So we need something that can be trainable in a reasonable amount of time and easy to deal with data because the PDB is a huge mess. So we want to avoid that. And so our solution was to go for nanobodies. So nanobodies are small antibodies which have a constant frame region, and they vary in 3 loops colored here in different colors. So those loops are very, very different, and they are sort of ab initio folding problem, while the constant part is easy to predict. And so at first we started with modeling using CNN. So the first version was predicting the distances for CDR loops. And then loop modeling with Rosetta. This is similar to Deep H3 from Graylab. But then we converted this to end-to-end modeling. So this is what we did this year. So my student, Tomer Cohen, he was master student. He's a master student in the group, and he was inspired by the end-to-end AlphaFold concept. And he suggested to implement the same concept for an nanobody modeling. Now, with nanobodies, we can do it in a much simpler way, because we can align them on top of each other. And then our structure module is much simpler because we don't have to take care of transformational invariance. And the evoformer model that is in AlphaFold is useless here simply because there is no signal in multiple sequence alignment, and the structures are variable, because this is the whole concept of antibodies and nanobodies. So we start with the sequence as an input. It is encoded using one-hot encoding, and we have a simple network that outputs the coordinates. For training, all the structures are aligned. And so Tomer converted this project into course exercises. So he prepared all the data ready for training, and we give it ready to the students. It can, so it has to be aligned, but they implement the structural alignment in the previous exercise, so they can do it. And then the network is relatively simple. We can use the ResNets, convolutional neural nets, and it is trainable in minutes, so the students are not frustrated with long training times. They can work in Colab notebooks. We give them a short introduction to Keras and TensorFlow, and then they compare the results to AlphaFold 2 on the same nanobody structures. So now I'll tell you a little bit about the hackathon that we do. So it's 2 days. As I said, we have groups of 3 to 5 students, and they can choose which projects they do, and they can continue from exercises or suggest their own projects. And surprisingly, most of them choose to continue with deep learning exercise. So here you can see students at the beginning of the hackathon or at the end when they are hungry. And they actually present a very interesting project. So this year, one of the projects was adding accuracy prediction to the network they implemented as an exercise. And you can see that they succeed quite well. Another project was the design. So given the structure, let's predict the sequence. And this was a group who had done introduction to deep learning course before, so they could implement interesting architectures, and they tried several of them. We tried to do multi-conformation structure prediction. So here you can see several conformations of the loop predicted by the network. Application to a different system, such as protein peptide that sort of has a fixed scaffold, and this concept can work there. And so they have 2 days, and at the end of 2 days, they really have all the code, and they even complete the documentation and the project report. So it's like a mini research paper, which is amazing to see. And it's great to have them say like, wow, we couldn't even imagine that we can do so much in 2 days. So I think this format is very successful, and we definitely keep it. And we will introduce more of deep learning in the future into the course. And so I'll finish here. Thank you.
Thank you very much, Dina. So now we can move to the Q&A session, if everyone comes back online. Yes. So I think one maybe comment to make. So I think, Dina, you have quite an advanced course, and it's clear that you are targeting a different audience. So computer science background, I don't think we would be able to ask for chemistry students or molecular life science students who do the same thing. So that's so you have to tune your course to the topic basically. But there was a question from Adrian, many people, should we teach our students about what is beyond those deep network or not? Probably Dina, you would answer yes. I would answer probably not. People are using Google without knowing how the search engine works. We are doing sequence alignments without knowing how the sequence alignment works. So there are many tools that we are using without understanding the details of how things are happening under the hood. So, Panos, you're teaching biology students, you probably don't want to start explaining them the network architecture. So I don't know, what's your view?
I'm also not the best person to explain this, like if they should do this, then Dina is the best person, like computational, more computationally oriented to the theory should be explaining these things. So a teacher has to know the subject that they are teaching. So I would not feel very confident I can read it and understand it, but I will never be an expert in this kind of very complex algorithmic structures,
I would say we have to teach them enough so they can understand the input and the output. For example, they have to understand that they need the multiple sequence alignment with sufficient number of sequences. We want them to understand how to interpret the accuracy prediction from AlphaFold. So we do want to give them a little bit more than just the black box.
Yes, which is again perfectly fine, and we should be done in your case. I think if they can do a hackathon and program their own network, then they deserve to understand things. So there's a question which has kind of disappeared from the list as being answered, but I think we need to talk a bit about it. And there are actually two related questions. So Juliette Martin asked the first one, should we still be teaching homology modeling? And then someone else asked, should we still be teaching docking? So Panos, maybe you want to kick off the discussion here.
Yeah, so for homology modeling, I believe that it's extremely important for students to understand that protein structures are similar to each other and how similar they are, and also the threshold. It's not only historical teaching, but actually it has implications, because if you have, like, for example, very high identity and you produce a homology model, I have been informed by Joao, who is a friend of mine that is working also on this kind of high-end homology models, that they are still useful for drug design. So they are using homology models for structure-based drug design. He works in a company in the US. So I think that homology model, both from a perspective on understanding a relationship of protein structures and for the application of like drug design in the future, it's important. So this is my opinion of this.
Ezgi, any views?
I also think that it will be still valid, especially considering what Panos has said. For example, there are very flexible proteins which visit different conformations, but they are quite distinct from each other. For such a case, you only get a single prediction currently from AlphaFold or from a related approach. But if you know what you want to have, what you expect, then I think it's still pretty valid to use homology modeling. And within that context, I think it will be for some more time still valid to teach homology modeling as well.
Dina?
Actually, yeah, I agree with both of you. It is still important, and it's probably one of the major challenges, getting more than one structure out of those deep learning models. But I have to admit that I don't remember when I used homology modeling last time, because somehow the first thing we do now is try AlphaFold, and I usually we stay there.
Yeah, so well, I remember when we last homology modeling last time, which was in the GPCR docking competition last winter. And we generated, so there the question is, so this brings us to drug design, I guess, what Panos was saying. So here you have to select conformations for your modeling that are relevant for the, the drug or the peptides that is binding. Is it an agonist, antagonist? There are conformational changes associated with that. And since everyone else was anyway going to use AlphaFolds, we say, okay, let's be stubborn and use the classical approach. So we did classical homology modeling, and we did get the best model for one of the 5 targets and failed completely on the other 4. So still a lot to be learned, but so I think this kind of aspect will still be important. And at least understand, how homology models are obtained and how to especially assess them, which part are reliable and not. So there was a related question, which was about docking. So do we still need docking or not? So I can probably start discussing that. We are still teaching docking. We're operating a web portal, Haddock, which is used a lot. We have not seen a drop in usage since AlphaFold has been appearing. So actually, during COVID, the usage has more than tripled the portal. So people still rely on docking. There are many things that AlphaFold cannot do in terms of docking. Maybe one day it will be there, small molecule. I heard them talk that that's their next target. Mutations are also the next target. Nucleic acids, RNA, they are not there yet. And one thing, so Dina mentioned integrative structural biology. So what AlphaFold cannot yet do is integrate data. I know that there are groups that are going under the hoods of AlphaFold to try to allow that, but think of cross-linking data from mass spectrometry. This is kind of like contact predictions from sequence alignment. So if you can start integrating a bit of data when you have it with AlphaFold, maybe that's going to be really interesting in the future, and that's probably going to come, will be my opinion. But I think for the time being, docking is still being used, and it's probably as homology modeling will still keep to teach those things. At the end, all those techniques are based on optimization algorithms, and understanding of structure, being able to analyze structure, energetics, and all of that. So I think this will still be interesting important to teach also. All the aspect of free energy, all these methods give you some kind of score energy. It's not the free energy, it's not the binding affinity, as Panos knows very well. And AlphaFold is not yet predicting binding affinities when it comes to, actually it seems to be doing quite well, because there's work showing that it can predict if two proteins are interacting or not, and some other tools are not doing that. But so should we still look into docking for the time being, or, I can retire and I'm probably fine, but the younger people might have a problem here. So, Ezgi, Panos, Dina.
So I think there's this technical bottleneck that you were mentioning in your presentation, Alex, that when you want to go for large systems, then using remote resources, web servers is not feasible anymore. Unlike the other docking programs, in many docking programs, you can input many molecules, 10, 15, can go higher. I mean, in the end, you can still use a web service. You don't need to install the program on your own machine. So that is also, for now, a technical limitation to use this tool in teaching during the class. Maybe for research, it might be different and we can discuss this, but for teaching, I think docking is still relevant.
Anyone wants to add anything? If not,
I would like to add something. So basically with docking, you don't get one solution or like 10 solutions. You can get like as many solutions as you want according to the problem that you are inputting, which means that it has also the concept of ensembles. And the concept of ensemble goes to dynamics and conformational states. So these kind of concepts are not yet integrated in AlphaFold. I don't know who is going to integrate it by including like long molecular dynamic simulations in improving predictions of AlphaFold, etc., etc. But currently, docking also gives you like an ensemble of solutions, which could be related like to maybe like on rates or these kind of things, like how proteins start, let's say, interacting. So you can discover not only the binding mode, but also like surfaces that are attracting the proteins to interact. So you can go deeper in understanding your protein-protein interactions.
I agree with both of you. I think docking is still very useful. So I will add that there are systems that simply don't work with AlphaFold multimer, such as antibody antigen. So those need to be resolved by docking, and it's also challenging with docking. host pathogen interactions. Basically, if you cannot get a reliable signal from multiple sequence alignment, AlphaFold will have a hard time putting it together. And larger systems are also challenging with AlphaFold. It still has the limitations, not only on the actual size, but the optimization there doesn't work well for large systems. So sometimes we do it by parts, or for example, we can take cross-linking data for a large complex, figure out by the cross-links which subunits have to interact, and try to sort of collect it or assemble it step by step using AlphaFold and docking approaches with crosslinks. So I think this is what is happening now, but I would say this might change, and we will see what happens in the current CASP, because there are many more groups who try to work on assembly question docking.
Yeah, I think the current CASP CAP3 running now will be very interesting to see if, other groups have closed the gap. There was a tweet yesterday, I think it was yesterday, about a method that can do the prediction from single sequence only. The preprint is not out there yet, but which seems to be doing even better than AlphaFold. So we have to wait for the preprint. So a lot of things are happening. So these are exciting times, and things are changing constantly. But I think you still want to, in terms of teaching, I think students should still understand some of the principles and some of the methodologies. And we'll see where things go. But I don't, at least in our place, we are not scrambling at changing our curriculum of what we are teaching. So that's pretty clear. I think if you're in computer science, maybe the things are moving much faster there. So that's for sure. So yeah, so homology modeling, docking, I think we have discussed. There's someone asking about courses connected to AlphaFold modeling. So there I don't have really answers myself. I see that, well, Dina, you seem to have a very nice setup in your course. I don't know if any of that is available online. That's maybe something for the EBI here to try to collect actually resources around all the tutorials and things that are there around this kind of artificial intelligence. Maybe it's there, but like a community resource.
No, it's not there, but I think this is a start, and hopefully after this we can plan to create some training portal with all the different materials, and I'll approach Dina to see if we can access some of her material as well. But anyone else who has useful information, it will be really useful for us to know about it, so we can collate things. I think there is so much to do in terms of collating training materials that we want to avoid doing the same thing again and again. So
yeah, so Elixir maybe also is a way to go, because they have this registry for both software and training material. And at the end, what you would like to have probably is kind of a voting system, or people can rate the quality, because at some point there will be hundreds of thousands of training material out there, and where do you start with which one is reliable, which one is not. So I think that will be important as well. So then let's move into disorder. I'm looking at the upvoted questions. So teaching about disorder. So I think someone was referring to the comments that I made, that I think my comment was more that it visualized to some extent disorder, because what is the PDB? If it's disordered, you don't see it unless it's in NMR structures, because NMR people typically put the entire sequence they calculate, even if they have no data, then you might see some of the disorder. I think Panos, you already answered a little bit. You might want to explain something, that there seems to be quite some nice correlation between what AlphaFold shows as spaghetti, or low score, and real disorder. You want to say a few words there, Panos?
In general, we have to evaluate this kind of scores on a case-by-case basis. So obviously there will be sequences that fall in different structural elements upon binding. So this kind of theory has to be covered at the master's level in order to teach this kind of disorder or flexible regions of AlphaFold models, because yeah, some of the questions say that like these are IDRs, but these are basically sequences that are flexible. So we don't know if they have IDR like properties, or when these kind of IDR properties appear, and they could be like molten globules or whatever. So for me, it's very important to like at least see this kind of flexible regions and visualize the models, as Alex and Ezgi said, because for me, it was very impressive. Even for me, when I started seeing these models, I said, okay, wow, like I learned something about like the protein universe, you know, as a structural biologist. So the overall answer to this is that, yes, we should take everything critically with a grain of salt on a case-by-case basis. But to me, this kind of non-prediction, of course, it can stem from a lot of factors. But anyways, it is there.
Yeah, and I think it will be important to also, you know, make students realize that disorder or flexibility, whatever you want to call it, is there. We have experience with our web portal, the Haddock portal, where we have people taking AlphaFold model and giving the full model to do docking, you know, with all the spaghetti around it, and then they don't realize, they just take it straight and that's it. So this is wasting computational resources. This is causing trouble, and it's maybe just a naive approach of trusting structure. But so that's probably even more important now when we teach structural biology, to also focus on the intrinsic flexibility and disorder, because people are going to use those models that are in the EBI database or use AlphaFold to generate them. And if they start running molecular dynamic simulations or do other things with them, and not realizing what are, say, reliable regions and disorder or flexible regions, they might waste a lot of resource doing their work and be disappointed in the results at the end. So, but I think we have now, I think it's the first time there is really a method that kind of at least visualize unreliable regions, because before we tend to not visualize them. But then you have to know that you should remove them if you want to do something serious in terms of modeling after that.
Yeah, so like I think that the IDR is like very, very important to discuss, because yeah, it was also not covered by the curriculum in general for teaching. For example, in Halle, it was not covered, and that's why like this kind of graduate school that we have the first like 13, 14 students that are going to go doing their PhDs by like working on disorder. So I think that AlphaFold also catalyzed this kind of education for them, even if these regions might not be as Vsevolod Filuga says. They're not disordered, they are just like ordered, but there is no structural homologue or whatever. But from what I know is that even if there is no structural homologue, AlphaFold can produce, like in one case, for example, that we had in the lab, a folded model at least. So I don't know. I think that at least it's very good for teaching, provided that the students already know how to assess. They have the theory, the physical chemical background that you taught to them about like disordered regions, unfolded regions, flexible regions, or disorder to order transitions, this kind of things.
Some people also have very specific questioning about there are different versions of different notebooks, which one to use. This is not per se teaching related, but so personally, I like I think the one, you know, Oskinikov's GitHub repository. DeepMind had its own one. There's probably more coming, but I think Oskinikov one has been very well maintained, updated. You get the old version, so that will probably be the starting point for me when looking at resources. But people might have different. And then we have a lot of the more smaller question there. An interesting one, you know, with cloud computing and open data and open algorithms, is it possible to do a PhD in an online manner? It's kind of this is education and teaching. My answer here will be probably that the last 2 years, yes, you can do a PhD in an online manner because we were all in lockdown and we're working from home. So, you know, if you're doing computational work, then you can work anywhere as long as you have the network. So that will be the online aspect. But you should not I don't know if there are universities that are providing purely online computational PhDs. That will be a bit strange. I think part of the training that is important when you do a PhD is also learn to interact with other people, learn to exchange ideas, and that's is not the same if you were to do it online. So I started traveling only in the last months, and I've been to 2 summer schools, and it's so fantastic to see students again. Everyone is so enthusiastic, asking questions. You know, it's nothing like an online school. So there might be things, it might become a commercial model. I'm not sure if you should give too much value to that. I don't know. So I don't see that happening. Following course online, I think it's fantastic. You can, there's all kind of online course where you can improve your skills, and that's very valuable. PhD online, I don't see it happening that quickly, but maybe others have different ideas.
Well, I think during the pandemic it worked because everybody was working from home, so it's a different setting and different motivation. But
you are still working in a group, I guess that's the thing. So the basis. Well, there are people who started purely online their PhD in the last 2 years, and then
sure.
Yeah. So,
as long as everybody is aware of it, and then the group is also mostly online, that person will be supported, I guess. But in a more, in quotation marks, normal setting, it would, I think, indeed be difficult to learn from the others, as well as not from the team members, but from other teams as well, because the setting is sometimes as important as your PhD team.
So one interesting one, we touch a bit on that. So this is becoming not per se teaching related, but so it's Vassam asking, since AlphaFold is not yet capable of predicting conformational viability, there is work in that direction. So there have been some preprints doing that, ensemble of conformations, metamorphic proteins. So these are proteins that can exist in different folds. Will we need different algorithms to predict the same? And what is a good place to start if you want to write your own deep learning algorithm to predict protein structure? That's a tough one. Maybe start following Dina's course and do the hackathon there, because that's what I think this is probably more the computer science part of things. Although I don't know, in my expertise also in teaching, you need to understand the biology, you need to understand the questions. I think probably some of my best PhD students had a background of training, in chemistry, biochemistry, and then they like to program, they like to deal with computers, and that's what gives some of the best results. And sometimes you have, for very complex projects, probably, if you want to develop software, maybe, studying computational science will be very important, because you understand things and you do better, but then you also need to answer the questions that you want to address them. Maybe we need a different type of education in the future. So how intertwined are computational biology and computer science in your place, Dina?
So I guess I'm lucky to be in a computer science department with a very strong group in computer vision. And I think computer vision is very close to us, because it's kind of similar problems. And so when we can define our problem in protein structure prediction very well, we can talk to them and they help us to find the most suitable network architecture. So I think this collaboration with computer vision or even geometric deep learning people works very well for us.
Yeah. And I guess not every department or not every bachelor or master program has this kind of connection. So that's, and it's not always easy to establish them. So that it depends very much where you are, I guess.
Yeah.
So maybe a general question to you, and maybe, some people in the audience might also comment on that. So we are all using Colab now to do some of these AlphaFold predictions, and also for teaching purposes. So what is missing? Is there something you would like to do with students that you cannot do? We are limited in our things. So that's more for, what should we give to EBI as task, or to the AlphaFold people if you want to improve teaching? What are we missing there?
Colab Pro account.
Sorry?
Colab Pro account.
Colab Pro account. Okay, so yeah, that's probably a business model there. Well, Colab education maybe. Something, yeah. But there are things, Ezgi, panels that you would like to do with students that are not possible to do? You think?
In the case of multimeric predictions, maybe it could be an idea to also have a resource, not as extensive as the monomeric prediction, like protein structure prediction version. It's still, I mean, these methods in general for multimeric prediction is still under development. But at least to have a resource so that we could already show the outcome without really waiting to have the result. That could be practical. For a subset of cases, even I think that would work from different systems or from different organisms which are biologically interesting.
You mean pre-computing?
Yes.
Okay, yeah, I agree. That would be very useful because many of the proteins currently in the AlphaFold database are not even monomers.
Yes, yeah, exactly. So if somebody has already calculated it, I think it would be also a good idea just to offer it. I know, I mean, it's a lot of work in the end, and also it needs storage space, but maybe a subset or a selection of it could be an idea.
So maybe a question.
I don't know, like for me, sorry, Alex,
go ahead.
For me, for teaching, like the AlphaFold database, for me it's enough, and the Collab Fold, and now the new tool that the Collab team developed, which is like extremely fast to integrate like into visualizing like primary, secondary, tertiary structure, and for the students, I think these are enough tools for teaching. But now for the PhD student level, that the PhD students want to do stuff, of course, you serve them on a case-by-case basis. When they come to the lab, you have AlphaFold installed on your servers. So I think this is for me, okay. Now I had other discussions with other people that want to like predict interactions, as Ezgi said, in a global manner. Like, for example, all the interaction, like the complete interaction networks of organisms with AlphaFold. Of course, there are, I don't know how this could be included in teaching, but you need the EU grid infrastructures to do this kind of calculations.
Yeah, probably HPC infrastructure as well, because of the size of the model that you need. So it's not so that's one of the limitations. But that's more a research question. I think the infrastructure to run large scale, I think that's. Yeah,
but for teaching, then the infrastructure is enough. Now, if you want to teach, to have a course on teaching visualization, 3D visualization of networks and interactomes, then of course you need this. So it's of course a matter of research, but it will also go into teaching, right? At one point.
Yep. So maybe that's a good, since you mentioned visualization. So there's also a question here about virtual reality. The question is more general, so it's not per se about AlphaFold, but you know, is there value to including virtual reality in teaching? It's very cool. We do it. So we have a virtual reality setup. We use Unity-Mall, which is developed in Paris in the group of Mark Baden, and we use it for docking contest for prospective students. So when we try to attract our chemistry students, they can dock in virtual reality, which is a very hard exercise to do, actually. But it's fun. And sometimes also for students, you know, to dive into structure. But for the time being, we use it ourselves more as a fun tool, but also dive into structure. So see a protein from the inside. I don't know if you will realize that instead of having an L amino acid, you have a D amino acid in virtual reality. So that would probably require a lot of training. But I think it's a way of attracting students, but I don't. Do you have any? Well, probably in computer vision, so you must be doing something with virtual reality on your side, Dina.
I think we took them to the virtual reality room once because we also have this setup. But in principle, it's only for I guess advertising purposes. You cannot teach them much from that. I do bring them 3D printed models to the classes, so I guess it does a similar. It has a similar effect.
Yeah, I think also the good old printed 3D models. Well, Ezgi knows about that. We gave that to participants on the EMBL course.
Yeah, I also.
It's also a nice way of showing the difficulty of the problem.
Yeah. And I guess I'm now in the EMBL course, so we had a similar conversation here yesterday. So I think where it does actually help is in fitting structures, even AlphaFold structures, to 3D density maps. Because there it's probably much easier to do it in virtual reality compared to using a command line interface of programs like code.
Mm-hmm.
At least that's what the modelers claim.
Mm-hmm.
So I guess there are problems where it can be useful.
So there is a question from Gerard Kleinwert, who is actually the head of the molecular structure cluster at EBI. And he says, well, most of today's discussion has been about teaching and structural and computational biology students. But as the models are becoming available to all, people in other fields, genetics, medicine, will have to learn about 3D structures and how to interpret them and generate hypotheses. So that's a very valid point. So, has any of the panel members begun to think about a plan for this in their institute? So basically, you guys are causing the problem, and we're asking us to solve it. That's what I'm reading from Gerard's question. So the problem here is that on my side, so this is my answer. So I'm teaching in chemistry, molecular life sciences. I'm not teaching in medicine or psychology or whatever in other fields where people might start looking at those models. So that will be, and I have no saying in what's happening in the teaching in other faculties. So that's, so hopefully people will become aware of that and will start to teach structure. But I think resources, so now I'm going to put back the ball to the EBI. You have your knowledge-based PDB knowledge base where you are integrating everything, and this is geared towards presenting the structural data to a very wide audience. So I think that's maybe the place where in a knowledge base, there should be clear indications on how to assess structures and maybe information there. So it will be hard to teach about structural biology in different faculties or different studies. Because if you are in a study where you don't think at a molecular level, so in chemistry, we think in terms of molecular and atoms, but if you have never been taught to think in terms of at that level, and you think more at the cellular and organism level, then it will always be difficult to look at those structures and realize that there are problems. So that will remain an issue. I don't know what the solution will be for that, but I think the way you present the data will be very critical there. Maybe that some of the others want to comment on that.
So as I mentioned, I actually teach in the molecular biology and genetics department, and most of my colleagues there from that discipline. And I actually went through all the hardship that you were anticipating, Alex. Now it's after 5 years, it's much better. So I know how to reach out to them. I know which type of jargon to use. But in the end, besides all the things that Alexander mentioned, and I think they're all valid, there is also a very positive thing in teaching people with genetics background structures. Because even when they design experiments, you see that they, of course, this is how they are trained, but they look from a very macroscopic perspective. And when they start learning about structures, you start realizing like the way they think about their research also starts to change. And I find this very valuable. And the more accessible the structural landscape will be, as I was also mentioning, the more time there might be left to teach these people really theoretical background in structural biology. And then we have been talking about this for many years, like merge of many different disciplines, like from macro to micro scale. And then when we start to talk in each other's jargon just a little bit, then I think this will turn into a reality. And talking to genetics like omics or medicine people, I think it has a really fundamental position within this landscape.
And I think, it depends also what people are doing at the end with the structure, because you might not need to understand everything, but if you take the structure as the truth and that's the end of your path, then that's probably not the way that people are using it. So I think structures but also experimental structures are good to generate hypotheses. And with those hypotheses, people go back to the lab, and they can test these hypotheses. So I think that's very important to teach also people that the structure is, even the experimental structures, there are issues, there are problems, and we might all be fooled. So I think it's probably more important that people realize the value of 3D structure rather than they are able to figure out that there is a problem in a very specific part of the structure. But I think that the database that are providing the structure will have an important role. And so with the worldwide PDB, every site can present information. So they rely on the same database, underlying database, but they can present the information in different ways. So I think in the way the information is presented, it will be so there's a role there for EBI and the other sites that are presenting structural data to directly provide some valuable information about the quality of the data. It doesn't mean that people will read those. So that's a challenge, I think, to make things visually appealing, that people cannot miss this information. So you don't want a long text explaining everything that is wrong, but so that's a user experience problem also. Or how do you present this information?
And also another aspect to Gerard's question is basically that there are the graduate schools at the PhD level. So graduate schools have a scientific question, and different kinds of scientists, they have their students, like they're coming from genetics, from cell biology. So they are in the graduate schools, especially for the disorder graduate school that we have here. It's obvious that they learn because there are also structural biology students. So whenever, for example, a student from cell biology presents a model and from AlphaFold, the other students are saying, where did you find it? Why do you show it like this? So it also, the training also happens inside the graduate school. Now, for the master level or the bachelor level, I don't know there how it can happen, but at the PhD training, the graduate schools are important, right? So these are the ones that are bringing people from different disciplines together.
So I think there's an opportunity here also, a training opportunity to develop, training courses, call it workshops, summer school, winter schools, targeting a different audience. And I know that at EBI, you have every year this structural bioinformatics course. The focus is shifting more to AI there. This is maybe more targeting starting PhD students in structural biology in general, but you can probably gear some new training courses that are directed more to medical doctors or other fields, and then you need to reach out to those. But I think there is a training opportunity, maybe indeed more what you say, Panos, at the graduate level or postgraduate level even. And this might not be something that you need to put at a very deep level in a bachelor and master education, where people might go into very wide directions. So what students do after they have studied chemistry, they might change completely fields. So you need to move to probably cover the fields that are defined in what the curriculum, what should students know at the end of their education. And maybe machine learning and all of that will be embedded more in different bachelors. But training people at a later, lifelong learning is a big thing these days. So maybe that's where part of the training should go, especially now because AlphaFold is out there. Everyone speaks about it. So we should not wait too long to do this kind of training, but you need to have the capabilities and resource to do that. And that's probably not going to happen at university levels. And EMBO, EBI, some of these organizations that support training and courses might have a role here. Samir, you want to say something about it?
No, I think I just want to point out we are almost close to the.
Yes, so we should.
And I think this has been really useful discussion, and many points that have come up. And again, I would say, if you have training material or if you think there are gaps, please contact us, and we want to bring all that together so we can create a nice portal for all the training material. And I just want to then thank you, Alexandre, Panos, Dina, and Ezgi for your contributions and really nice discussion. And thank you to all the participants.